So this Friday we had two very interesting brain metastasis studies. The first study was a study in non-small cell lung cancer patients with an ALK translocation, the ASCENT-7 study. This was with ceritinib. And interestingly, in this study, only patients with active brain metastasis were included. Also performance status 2 patients were eligible, patients with stable neurological symptoms and patients with leptomeningeal metastasis. And patients with leptomeningeal metastasis were discussed in another session that day. Based on prior brain radiotherapy and prior ALK inhibition, patients were divided into 4 cohorts, and all these cohorts were treated with ceritinib 750 milligram fasted. Based on the cohort, so approximately 40 patients were included in each cohort except for one. The objective response rate was around 30% in the brain in those that were pre-treated with ALK inhibition, and around 50% in those that were untreated with another ALK inhibitor. Disease control rate in the brain was very high, was above 80%, and duration of response was 9 to 12 months. And if you compare that to the ASCENT-4 and ASCENT-5 data, I think results are very similar and very promising in this setting. So how to put this into context? Well, we also have the ASCENT-8 data, so ceritinib 450 milligram with food, that results in less adverse events. But if you look at the PK data of the ASCENT-8 compared to the ASCENT-7, to the 750 milligram without food, then PK data are similar. So you would expect that with ceritinib 450 milligram with food, you would have the same brain results than 750 milligram without food. But it would be very interesting to see these results from the ASCENT-8 trial. So how to move forward? I think, well, we need to be aware that we need to design clinical trials for this specific patient population, so lung cancer patients with brain metastasis. We need to have bicompartmental endpoints, so you need to define what to do with the brain and what to do with the extracranial disease sites, as it is possible that some patients respond in one compartment and other patients only in the other compartment. We need to evaluate whether TKI concurrent with cranial radiotherapy are safe and effective, and whether it can prevent a disease flare. And I think we should focus on the next generation ALK inhibitors that have high blood-brain barrier concentration. But I think it's very difficult to compare all these trials because inclusion criteria were very heterogeneous across all trials. But in general, they are all very promising. So moving on to the next abstract, we know that brain metastases are associated with a lower quality of life, and it would be very interesting whether we could predict which patients have brain metastases and which patients have not. So in a small Asian series, patients with synchronous brain metastasis, 18 patients, and patients without brain metastasis, still dead, 31, were included. They had matched tissue of the brain and the primary, and the authors evaluated whether molecular alterations could predict which patients had brain metastasis, but they were not able to do that. Interestingly, they did also methylation analysis, and with certain methylation blocks, they could very accurately predict which patients had brain metastasis and which patients had not. So how to move forward? Well, I think this should be validated in larger series perspective with extensive information on staging. But it would be the best if this signature was also predictive for development of brain metastasis in the future. So in the end, that you can design trials which really aim at predicting brain metastasis development in high-risk patients, for example, with prophylactic cranial irradiation or with epigenetic drugs.